The next speaker is, sounds cliche, but really does need no introduction. And you may have noticed that Dr. Bonneman's name came up numerous times last night, and that's not a coincidence. Dr. Bonneman is a pediatric neurologist specializing in neuromuscular disease and neurogenetics. He is a senior investigator in the neurogenetics branch and chief of neuromuscular and neurodegenerative disorders and of the childhood section at NIH, NINDS. He is also a member of the Foundation's Scientific Advisory Board. We are extremely lucky to have Dr. Bonneman with us this morning to give an overview of RYR-1 related diseases. I cannot think of a better person to do this literally in the world. We are very lucky to have Dr. Bonneman. He has diagnosed so many people with RYR-1, people who had gone to many, many doctors with very challenging circumstances where they weren't getting good answers, and then they show up in Dr. Bonneman's clinic and things get cleared up right away, and that's a huge sense of relief for families. I don't need to tell this group about that. So Dr. Bonneman, the floor is yours.
Thanks, Mike. So, good morning. Sorry, I have a little bit of a hangover from a virus. It's already 2 weeks old, so it shouldn't be infectious anymore. But what I'm going to try to do this morning is really give a high-altitude overview over RYR-1 related disorders, really as an introduction to all the talks that will follow this morning, so that we are all on the same page for this. And even though I'm a clinician, of course, we've seen a lot of conditions, including a lot of patients with RYR-1 related myopathies. It's not my primary lab research focus, but we have the privilege today of having many expert scientists in the room as well, who will forgive me if I simplify things too much and can then make up on it when we talk about the details of all of this. So I want to just make some high-level comments to really put this into perspective, because I think it's very important to know what we're dealing with. We're in muscle disease in general, we're dealing with something that many other conditions don't deal with, which is an extremely large organ. Muscle, if you understand muscle, skeletal muscle as an organ of the body, then it's one of the biggest organs that we have. And depending on your level of fitness and body mass index and so on, it's actually 40 to 50% of body mass in people that is muscle. And it's not only one thing, it's over 600 individual muscles that the body has. And all of these play some roles in the function, from mobility to expression to pure survival, the respiratory muscles, for instance. So it is important to try to get to as many, if not all of the muscles, because all of them will, as you will see, require RYR-1 function to properly work. So with that said, what does muscle actually look like? And here again is a fairly simple diagram of what it takes for a muscle to execute a movement, something that you can actually see or that you make use to walk or point. So we have a bone, and the muscle needs to attach to the bone with a tendon to be able to transmit force. And then the muscle is composed of these muscle fibers, which are very interesting cells. They're actually cells that are very long and have many nuclei in them. They call them syncytium. And normally a cell is, one little kind of component with one nucleus in it, and it is many of these different cells together that make a tissue. In muscle, that's different. In muscle, we have a long continuous cell that has many, many nuclei in it, and that happens because reserve cells, the satellite cells, fuse into it and contribute to this big cell. So that's what this looks like, and it is composed mostly of this down here, which is the contractile apparatus, which is what makes muscle contract. So the purpose here is really to shorten all of this so that it pulls at the tendon and moves the bone, right? That's how you create a movement. And so that happens on a microscopic scale, a millionfold over of all these what we call sarcomeres, things in register that contract all at the same time, and then the entire thing is visibly shortened and you create a movement. Now, for that to happen, you need to excite it. So you need to kind of make it contract. And we come back to that. That's where RYR-1 comes in. So remember this, this is the sarcomere, this is what makes muscle contract, but you have to make it move and contract really to make a movement happen. So the other concept that is important to understand when we talk about RYR1-related myopathies is the concept of a congenital myopathy. So that's the umbrella term of RYR1-related myopathy. So what do we mean by a congenital myopathy, and why do we even use the term? So these are some very basic concepts what defines a congenital myopathy, where the RYR1 disorders in the broad sense belong. There are non-dystrophic genetic muscle diseases. What I mean by that is, you know that there are muscular dystrophies, which are degenerative muscle disorders, where the muscle is fragile and degenerates and pops over time and degenerates because of that. And those are progressive disorders of muscle. We call them muscular dystrophies, and you're probably aware of Duchenne muscular dystrophy as a paramount disorder. So the congenital myopathies are different in that they're not primarily dystrophic or degenerative, and we come back to that with some caveats later. And congenital in that term means prenatal onset. That means in the classic presentation of many of these disorders, there are signs and symptoms of babies even at birth. So they're born with low muscle tone, maybe some contractures, some weakness. Sometimes things are picked up prenatally. Means when I had congenital, it doesn't mean the baby has been normal up to birth and then it's born and whoop, the disease is there. No, it is kind of something that builds up to birth and then they're born with it, and that's congenital. In congenital, you can stretch that. Sometimes the findings are so mild, or the affection is so mild, that it only is picked up when some motor milestones are delayed, or when the child gets into running and playing mode that the kid can't keep up with the other children. That's when the problem is recognized. It doesn't mean the problem starts at that point. It's just recognized at that point. Most often there's been some mild underlying muscle problem all along, but it didn't come to attention. Maybe the baby was a little low tone or things, but the pediatrician says, well, don't worry, it's going to go out of it. But then when the motor milestones are really have to kick in, it becomes apparent that there is a delay. But there is true onset later as well, and that's what we've realized for all the congenital muscle diseases, including RYR1. Yes, at the core, they were recognized as congenital muscle disease, but when we now recognize the entire spectrum of clinical cases that we know of, there are some that really start later, where the childhood is normal, and then really things become apparent only in adulthood. And still, it belongs to the big umbrella of RYR1 related myopathies. So the causes of congenital muscle disease are typically in that apparatus that makes muscle move, that kind of cellular apparatus. And that makes them different from a dystrophy where the problem is in the stability of the muscle fiber. And we'll come back to that because it's important. Well, classically, the congenital myopathies were classified by what you saw under the microscope, and that gave the disease its name. So here is core myopathy, which is, of course, something we come back to, which is this central core disease, which is the most common form of RYR myopathies. But the classic myopathies also include Nemelin myopathy that you may have heard about, centronuclear myopathy that we will be talking about, and something that's congenital fiber type disproportion. So those were the easy days some decades ago. And then genetics comes into play, and it turns out any of these simple names have many, many genes underlying them that can cause them. And I don't want to, well, there will be a test at the end, and you have to remember all the genes. But this is just to show that all of these classic histological names have many genes underlying them, and I've highlighted in red RYR1. You can see RYR1 actually plays a role in almost all of them. So the spectrum is much broader than just the core diseases where they were defined. And so we have to be nimble, we have to be more complex. So how do we define these diseases? Clearly by the gene, that's why we are all in the room, RYR1, but also by the histology, so what we see under the microscope, but also what the clinical presentation is and what the mechanism is as a result of the RYR1 dysfunction. And that all has to kind of play together, and clinicians have to keep all these balls in the air to do a single patient justice by understanding all these elements that go into the disease. So back to what the mechanisms are. This is a diagram that we put together some years ago to highlight again what congenital myopathies actually do to a myofiber. And I think the two most important aspects of congenital myopathies are here, which I mentioned already, which is the sarcomere, the contractile apparatus. Many congenital myopathies happen there in the contractile apparatus. Classic example would be Nemelin myopathies that you've all heard about. But the other very, very important component is this thing here. We call that the triad. And that is where calcium is released into the myofiber to trigger the contraction of the sarcomere. So that makes a muscle actually contract. We call that excitation-contraction coupling. The nerve up here sends a signal to the muscle, hey, you've got to move. Our owner wants to go right, we better move. And the nerve signal comes down, and then the muscle fiber recognizes the signal and sends an electrical signal down to this triad structure, an action potential. And that electrical signal opens this calcium channel, the RYR-1. The calcium bursts into the myofiber, the contractile apparatus says, oh, calcium is here, we better contract, and then it contracts. So that is the two parts of the excitation contraction where most of the congenital myopathies play out. And in the triad is where our home is, where this RYR-1 is. And you can see that here again in another diagram. Here are these tubules, these membrane guidances for the action potential to go deep down into the muscle and talk to the RYR-1 receptor to now open. The signal is there, open, release the calcium, here you can see it, make it contract, but then rapidly get rid of the calcium again, because if there's calcium hanging around too long, that's bad too, because then you get into sustained contraction, and calcium can also do damage to the myofiber if it's not regulated. So it's the homeostasis, the regulation of the calcium in the myofiber that is so essential to proper functioning of the muscle. So yes, you need to release it, but you also need to get rid of it right away after it's done its job. And if the channel is leaky and kind of oozes out calcium at a steady state without reacting to the signals, that's one of the reasons you can get RYR-1 myopathies. So what causes RYR-1-related myopathy? So clearly, it's the genetic variants. That's our new term, formerly called mutations, but the genetic pathogenic variants in the RYR-1 gene are at the start of all of this. But then it is the calcium homeostasis that is affected, and that's where things play out differently depending on what happens to the channel. And we hear more about that later in the day. But it is the critical component, start component for that excitation-contraction coupling. And the impairment of this balance of calcium regulation causes weakness or even damage to the muscle fiber. And so there are different ways to get there. That's something that we hear about in the coming talks. But this is when the calcium works properly. You can see how the stroke of the sarcomere happens. That can only happen if calcium is there to release the block on it. And this is what we're dealing with. This is the beautiful structure of the RYR-1 calcium release channel. 4 copies of the protein come together to form this tetramer. You can see here, that's a channel pore. That's where calcium goes out, and this is what reacts to the action potential here. Much more to come on this structure. So one thing that you also hear about a lot, and including when I go on with my talk, is we're talking about dominant and recessive genetic variants in the RYR-1 gene causing the disease. And Livia and Andrew will be talking about that much more in the next talk, but I just want to kind of lay the groundwork without going into too much detail, why it's important to kind of recognize that we have these two genetic mechanisms, because there is a difference in inheritance and also in recurrence risk in families. Dominant, you need only one pathogenic variant that causes a problem traveling down generations, and in the recessive forms, it's 2 that come together in the child with obviously quite different recurrence risks, as you will hear next. It also has consequences, as you will hear from Dr. Marty, that what happens to the channel as a result of these different genetic ways to get RYR-1 myopathy. And that results in clinical differences, as you'll see as we go forward. But there's also a lot of overlap. So it's kind of not a firm distinction, it's kind of a floating spectrum of disorders that come out when you have dominant or recessive variants in it. The recessive RYR-1 myopathies are newer to us than the dominant, and that's simply because of the size of the gene, which we could not properly analyze until next generation sequencing technology came along. And now we can, with genetic testing, look at the entire gene in everyone, and we discover these recessive variants much more readily. But the red sentence here is absolutely key. It means that you have to work with your geneticist providers, neurologists, to make sure that the genetic information that you have is complete to allow for a confident diagnosis of an RYR-1 myopathy. So that always in every patient has to be looked at very critically to say, well, yes, this variant is responsible, or this variant is only half responsible, I'm missing the second one, I have to keep looking, or this variant has nothing to do with my problems. So that's really, really critical. So we have one gene, but we have many possible manifestations of the disease, and those include the onset, as I mentioned, the distribution of the muscle weakness, the severity of the weakness, the associated pulmonary and respiratory involvement, orthopedic involvement, but also the risk for malignant hypothermia and rhabdomyolysis, which is sudden breakdown of the muscle that can also happen as a result of dysfunction of this channel. And we're not going to talk much about MH and rhabdo, but we can in the discussion sections coming up. That's a feature of some of the channel variants where the channel is just hypersensitive and will inappropriately release calcium into the fiber, causing this rapid contraction, which is malignant hypothermia, where the muscle goes into contraction, the temperature goes up, and the muscle can break down. And that can happen without a myopathy, but can also happen as a result of the pathogenic variant that causes a myopathy. So MH risk is always part of the genetic assessment of an RYR-1 myopathy to define whether a given patient has an additional MH risk as part of the variant in the RYR-1. And then there are non-neuromuscular manifestations that are emerging, more readily now, and these include metabolic changes, hematologic abnormalities such as bleeding, and also pain in some patients. So the historic perspective, and this is a slide from Payam, is that this disease was recognized by many neurologists and physicians way before this seminal paper some 70 years ago. Was it 70 years? Yeah, almost 70 years ago was published to really put this disease on the map. But they came up with all kinds of different names and didn't really know what they were looking at until this came up, this paper here. And Milton Shai actually was a clinical director at NINDS in my institute. So there's a direct connection to this. And he was really expert in identifying clinical syndromes and looking at the muscle biopsies very carefully. And what he recognized is that there were children with a congenital, so present at birth, but not very progressive muscle disease. And he recognized this was inherited as a dominant because he had multi-generation families where things happen in one generation after the other. And he looked at the biopsies and found these weird things here that he called central cores. That gave the name to the disease, central core disease. And I'll explain to you a little bit what a core is. And he saw that there were delayed motor milestones and lower extremity weakness in particular. And some patients also had stiffness of the muscles after exertion, which was also a very relevant and true observation at the time. So what are cores? Cores are not really holes in muscles. So you don't envision them as kind of someone punched a hole in the muscle fiber. They really show up when you stain the muscle fiber for mitochondria, which are the energy-producing organelles in muscle fibers. And when you do that, as seen here with this blue stain here, you can see there are areas in the middle of the fiber that don't stain properly, and they look like holes, like cores in the fiber. And when you look at them, this is a cross-section through the muscle, and when you look at it longitudinally, you can see they really extend the entire length of the fibers here. And that is a very peculiar phenomenon. And when you look under the electron microscope, what happens to the fiber where the mitochondria are no longer properly located, you can see that the sarcomere, the contractile apparatus that I described to you before, which is seen here as a striped tiger pattern in muscle under the EM, electron microscopy, you can see where the mitochondria are no longer there. It looks all disorganized now. It seems to be falling apart in these cores and no longer functionally aligns. So that's a core. It's this kind of mislocalization of mitochondria, and then as a result of that, a disorganization of the contractile apparatus in the middle of the fiber, which probably also contributes to the muscle weakness that we see. This is a fairly classic disease indeed. It is demonstrated here is an almost 2.5 to 3-year-old, and you can see she was born with some hypotonia, and she has difficulties getting up the steps. And her mom had also a lifelong problem walking, which was never properly diagnosed. They labeled her as CP and all kinds of things. But look at mom, and she's just the same. Just the same, but many, many years later, it hasn't really worsened from what you see in the little child. So that shows you that that is the non-progressive nature of it. When you look here at the fibers, again, this is a biopsy from this family. You can see the central cores that I showed you before. You can see the disorganization of the muscle fibers in the middle, and you can see there is some kind of protein detritus or gunk building up in the middle of the fiber, and the outer diameter of the fiber looks quite normal. Typically, these children have lower extremal leg predominant weakness, proximal predominant weakness. They may even be born with hip dislocation because it starts, as I mentioned, prenatally, and sometimes the hips don't stay in the socket because of the weakness of the fetus at the beginning. But it is quite non-progressive. There may be some spinal deformities, as you will see, and there may be malignant hypothermia risk. So with the advent of complete genetic testing in the gene, we now have a much, much broader spectrum from this classic core phenotype, no pun intended, the core phenotype of central core disease. And now we have this kind of huge, huge spectrum, and another Payam slide here that shows you all the different manifestations of RYR-1 myopathies, muscle disease that we now know. And it divides it here quite nicely into static problems and dynamic problems. Static is the congenital form that I just showed you of central core disease. It can be congenital, can be later onset. But then, as you'll see, there are also cases, particularly in the recessive, but also some of the dominant, are much more severe. So severe that it is sometimes not quite compatible with life at birth. But most of them are, and then you'll see that there is a particular subgroup with involvement of the extraocular muscles as well. And then we have the dynamic problem, which is the malignant hypothermia, of course, which is intermittent, but there can also be intermittent weakness, intermittent stiffness, intermittent muscle breakdown, and some patients have mixed features of static and intermittent, such as the King-Denborough syndrome, which is also an RYR-1 disease, typically dominant, where you have skeletal abnormalities, scoliosis, some facial differences, some weakness, but also a propensity for malignant hypothermia as a dynamic one. And I mentioned with the advent of next generation sequencing, we are getting now into the entire spectrum of the recessive forms that we didn't properly diagnose before, because we only looked at the hot spots of mutations where the central core disease arose or the malignant hyperthermia. And these are some of the early papers. You can see early onset children affected here. And you can already see in the early papers, some pointed out that there were restrictions of the eye movements in some of the patients, which is a finding that deserves another look as we go forward. We also at the time collected cases that were children that were quite severe on the RYR-1 spectrum with congenital onset, and put that out there, and it became apparent that RYR-1 is much more of a spectrum than just central core disease. These are children born with often contractures, severe weakness at birth, some with breathing and nutritional swallowing problems. But you can also see that some of the features overlap between more severe dominant condition and a severe recessive, in particular the spine, the scoliosis, which can be a real problem in the early onset RYR-1 myopathies and deserves great care. And some of the recessive children are also as very, very similar to the classic central core disease. So you can see this boy here. He would be very, you could say he's just like a dominant central core child in his weakness and his pattern and his non-progressiveness. So there clearly is an overlap between the dominant and the recessives, but the recessives tend to be more colorful in their clinical presentation, also in the muscle histology, and tend to be more on the severe spectrum. But it's also a condition of all ages. So this is Nicole Fuhrmann's paper where she, with Heinz Jungwirth, pointed out, no, well, don't forget, this is not only a pediatric disease, that is a disease of all ages, including adulthood, including patients with involvement of the extraocular muscles. I think this was a patient with rhabdomyolysis, if I'm not mistaken, Nicole, and this is a classic central core patient. And so this reminds me to say, well, yes, there is a spectrum, and we say, well, that's severe in the children and there's mild in the adult. That's not appropriate terminology, because in an adult affected with these conditions, the term mild is not appropriate. It can be just as impactful on daily life and activity and functioning as the more severe quote-unquote diseases in children, and in particular in adults who now live independent and have no support structure, and often are also not supported by the medical system, as we as pediatricians, we hover over our patients and families, and some of that gets lost in adulthood if it weren't for physicians like Dr. Fuhrmann, who recognizes that problem and really takes it on. I mentioned that, in particular, the recessive conditions now have a plethora of different manifestations also in the muscle biopsy. This is really not to belabor, but you can see central core, which is this one here, is just one of the possible manifestations of the disease on muscle biopsy that we now recognize. And so the biopsy is a supporting tool, but it's no longer a diagnostic tool in that way. So the muscle biopsy can look many different ways, also depending on where you take it. There's some correlation with dominant and recessive. So the recessive ones cover almost all of the possible histological manifestations, and the dominants are maybe fewer with the core, again, in the central core disease. But this is really not that relevant for the discussion. What is, however, an important, I think, observation is the involvement of the eye muscles. Really look at the eyes. And here are two young girls who are still friends, by the way. And you can see this little girl here has difficulties, even at this young age, to follow a toy properly. You can see she's moving the entire head instead of the eyes, so she has some restriction of eye movements. This girl here with very similar neuromuscular manifestations and degrees of weakness has completely normal extraocular muscles. Both of them have recessive RYR-1 disease. But one has a finding on the muscle biopsy, and that's why the muscle biopsy isn't completely irrelevant. That looks like centronuclear myopathy with lots of nuclei, muscle myofiber nuclei in the middle of the fibers here. You can see that here. And this child has more of a core phenotype in the biopsy here. What ties them together, interestingly, and helped with the diagnosis in both of these children is when you do look at the muscles that are affected, both of them in the thigh muscle have this curious sparing of one of the thigh muscles called the rectus femoris, which for us is a marker of this group of diseases. So distinction and overlap between these two phenotypes are very clearly evident. Why do I think that is important? Well, I think this phenotype here in particular, this clinical presentation of recessive RYR-1 with the eye movement involvement and with the biopsy looking like a centronuclear myopathy, ties this disease to other congenital myopathies. If we go back to this diagram and to the triad, it turns out the centronuclear myopathies as a group of congenital myopathies are also disorders of the triad. And these are the genes. You may have heard of myotubularin, MTM1, DNM2, BIN1, but also RYR1. So really, these two disorder groups, the core myopathies and the centronuclear myopathies, are really tied together, not only by the histology, what this looks like in the biopsy, but also by the clinical appearance of it. And we heard testimonial yesterday from a young man who said he was diagnosed with myotubular myopathy because he looked so much like it, when in fact he has RYR1. Why do I think that is actually a hopeful thing? I tell you that is a hopeful thing because in myotubular myopathy, which is a very severe congenital myopathy that really looks like the recessive RYR1 with centronucleation and extraocular involvement, there was a gene therapy trial, which unfortunately is on hold because there was toxicity to the liver, which is an entirely different story because of the virus and the liver disease that these children also have. But the thing that we saw in the children on the muscle side was really astounding. So even in a severe congenital weakness, things got to a point that children walked. Children who normally not even were able to sit and got off the respirator from 24-hour respiratory support to none. So that tells you, even in a severe myopathy, that's very related to what we see in RYR1. If you do an appropriate causative treatment, there is potential for striking improvement in these disorders. So that, I think, is a note of that we really should kind of take to heart and think this is worth pursuing. Now, RYR1 is a much tougher target. Do I have some more time? Or 5 more minutes? We'll do it. What is the cause of this myopathy over a lifetime? And I'm just going to highlight two studies just as a brief high-level overview. This is a retrospective study from London in children, pediatric cases with RYR1 myopathy. And basically, the two messages from this are there is some respiratory involvement that often goes unrecognized, that's slowly progressive over the years, that should not be recognized. And lo and behold, it's very hard to decipher this graph, but let me explain to you. This means mostly stability over the years. Some children did lose ambulation over the years. Some got better, in particular on the swallowing function. But mostly stability. And this is a study from adult patients from France that shows two things. One is that RYR1 is the most common of the congenital myopathies, as we now know, and also that in the pulmonary involvement, there's very few patients with very severe pulmonary involvement, but when it gets to moderate to milder pulmonary involvement, it's very prevalent in RYR1. And then secondly, most patients do walk, but there's a substantial proportion of patients that need walking support or even wheelchair support. Why can these conditions get worse over time, even though it's not a muscular dystrophy? Well, one is that the muscle is impaired by the disease, and it has less reserve, and it needs to do more to upkeep itself, and that can get difficult over the lifetime of a muscle. We heard in the scientific session that the satellite cells can also be affected by this, so there's a limit to regeneration and upkeep of the muscle, and often that becomes apparent as a patient goes into middle age, in particular with these myopathies, where some progression of the disease can be noted. On the flip side, in a child where the muscle still grows, the muscle growth can sometimes outperform the disease, and there's some improvement of it, and then a plateau, and then there may be some slow progression of it. So there are principles of care that are relevant to the RYR-1 myopathies that we will touch upon in the course of this and the next day, that are sometimes just general care guidelines for myopathies in general, and some have more, of course, of an RYR-1 specific feel to them, in particular malignant hyperthermia and rhabdomyolysis. But this respiratory involvement is something that is really important, because yesterday also we heard a testimonial of a patient who went into right heart failure because of underventilation of the lungs over time. So that's something to really keep an eye on. We hear from that from Dr. Meyer. And the second, of course, elephant in the room for the early onset cases is often very difficult to treat scoliosis, and we get to that as well. So finally, I want to just highlight that we also should talk about genetic therapies. I mentioned that for the myotubular myopathies, and so we should also think about it in the RYR-1 disorders. This is just a super simple diagram of the flow of genetic information. Our pathogenic variants, the mutations are in the DNA. The DNA is transcribed to RNA, that's made into protein and muscle, and that makes a human being. And in a genetic disease, all of this fails in a way, and starting with the mutation. And so our paradigm in genetic therapies is we have to get as close to the source of the disease as possible to have the highest impact that's possible. That's a challenge, as you'll hear in RYR-1, because the gene is so big. To just replace it and deliver it properly is difficult. We need to know what the individual variants in the gene are actually doing to the channel function. Do they act as a dominant? Is this a recessive? How do we plan our therapeutic strategies? You'll hear more about that soon. But we also have to understand and address all the downstream consequences that happen, that are touched upon, to the muscle contraction, to the muscle integrity, and to the reserve cell populations. Because in the end, I will tell you that no one therapy will be enough. You have to address it at all three levels. And that includes proper proactive care for the patient. That includes addressing the consequences of the RYR-1 dysfunction in muscle to make muscle more resistant to it. And then, of course, in the end, will include addressing the genetic abnormality. And that has to be tied together. This is what we already can do, the green arrow. We can do proactive care in these patients, and that lays the groundwork for everything that comes after that. And Dr. Marty will be talking about all of these aspects in her talk as well. And I will thank you and leave it with questions. Thank you.
Thank you. Macy.
Hi. I just had a quick question. You spoke a lot about muscle biopsies, obviously. We've been told that it's not really important to do a muscle biopsy to determine how severe my son will be. Do you agree with that?
Thanks for bringing that up. So I was hedging a little bit there, because I'm a muscle doctor, I like looking at muscle biopsies, right. And I like looking at the histologies, and I find it fascinating how sometimes the histology is helpful and sometimes it's not. But you're right. If you have a genetic diagnosis where the genetic variants really fit together and the answer is clear, you don't need a muscle biopsy at this point. However, sometimes, and that's why it's so important to be critical about the genetic findings that you have, sometimes you may see a pathogenic variant in the RYR-1, one of them, but you recognize this variant is actually recessive. So I'm missing the second variant and I can't find it. then a muscle biopsy may be helpful, because then you can look at how the RYR-1 gene on the other allele, on the other chromosome is transcribed. Are there any abnormalities that you didn't pick up on normal sequencing? Then it becomes relevant. So it really is, muscle biopsy now comes after the genetics. It used to come before the genetic,
Yeah.
now it comes after the genetic.
Okay, thank you so much.
The role of muscle biopsy for MH susceptibility testing I will leave up to the experts.
Do you find the progression of RYR1 get worse generationally?
No, I think so. That question. So in some muscle diseases, there is a generational progression of the muscle disease. So the most famous for that is myotonic dystrophy, where it kind of can jump from mild to severe, and there's a genetic basis for that. The mutation actually gets more severe in the next generation. In RYR1, we don't know that, but in RYR1, what we found is two things. One is that there is individual variability of how a given mutation plays out in individuals, and so even in siblings with or parent-child pairs with the same variant, that can play out differently. So that has to be kept in mind. That is just variability of the human genome that interacts with the mutation, and some are more resistant to it and some are more susceptible to it. Something, of course, we want to know. We want to know, why are some patients more resistant? Is that a therapeutic avenue that we can take? The second important consideration is, now it gets a little complicated. So sometimes you have a mild dominant mutation, a mildly affected parent, may not even know or just be mildly affected, gets passed on to the child, and then a second recessive variant comes in addition to that. And then the child will be much more severe than the parent. So that's another genetic scenario.
Hence my question, because that's our situation. So thank you.
You talked a lot about the calcium handling process with RYR1. Something that's been recommended for my son is albuterol, and we haven't started it because they told me it would improve calcium storage, but his defects in the pores, so what you said about leaky pores, yeah.
Question, I will punt it to the expert there.
So we're actually going to, I'd love to get, is Dr. Dirksen here? Can you comment on, we're actually going to have a whole session today about currently available treatments, including albuterol, but Dr. Dirksen, do you want to just maybe briefly sort of talk about where albuterol may be acting? Because I think, real quick, show of hands, how many people, if they're willing to share, are on albuterol? So a fair number of people are on albuterol. Okay.
Rational choice, because it does build muscle, it kind of builds neuromuscular junctions. But the question of what it does to calcium is an excellent one, and so we have to.
An unclear answer to this question, but there's evidence that albuterol promotes the ability of the store to refill with calcium. So when there is leak, it can help to refill the store. So under conditions of those mutations that promote leak, that this may refill the store, although again, this is not completely fully understood at this point.
That, like, you could store too much? Okay,
I don't think so, no.
good to know.
Calcium is leaking out through the ryanodine receptor that's not functioning. And the thought is that albuterol may help that calcium get back into where it needs to be. So too much is leaking out, and if we can do something to help the calcium get back into where it needs to be, which is called the sarcoplasmic reticulum, and Dr. Dirksen is going to be talking about that, that may be the mechanism of how albuterol is working. Go ahead.
Just to add that it's not without side effects, and I would always consult with your clinician before I start using it.
Hi, I'm trying to get my thoughts together, so I apologize if this question's a little weird. But our son has an unspecified RYR1 gene abnormality, and when we found out, we had all 4 of our kids tested for MH, and it turned out that our youngest has the same exact gene abnormalities as our son, but is not showing any physical symptoms whatsoever. And I'm just wondering, as you were talking about how symptoms can show up like later in life, or like, is there any idea as to why two patients would have, and their ultrasounds were the same with the muscle deterioration, why they would have the same thing but be demonstrating it very differently?
Yeah, so first of all, one has to make sure that this is this a VUS variant of uncertain significance? Yeah, so we have to kind of scrutinize that, and we can do that maybe this afternoon, first of all, to make sure that this is actually a candidate for causing the problem in the family. And if it is, and the one variant is sufficient to cause the disease, then one has to understand why there is variability. And so in RYR1, some variant, there is differences in what we call penetrance, meaning when the disease will surface in a given carrier of the same mutation. And that could be the explanation here. But one has to kind of step back and really scrutinize the variant, make sure that it actually is a fit for a clinical problem.
Real quick question, Carson. I ask this to you every 2 years, I think, but I think most patients with RYR1 would tell you that not all their muscles are affected in the same way. And classically, RYR1 is more proximal, hips, shoulders, but there's huge variability. So how do you explain, if all muscles have the same variant or the same mutation in their RYR1 gene, how do you explain the differential involvement?
Yeah.
My entire life, and I'm just wondering if there's any new updates on that issue.
Question, and this phenomenon is not restricted to RYR1, is actually typical for any genetic muscle disease that I know. Georgia has studying that for a living in his MRIs, and we kind of look at it and say, oh my goodness, this is disease X, this is disease Y, because the pattern is so typical. I don't know why that is, but you're right, the genetic defect, if you will, is the same in all muscle fibers, and you can study all muscle fibers and they will have it. So what I'm hoping for, that we'll be learning from that to see, well, how come that some muscles with the same genetic variant are so well preserved in a muscle disease? What do they have to make up for, to make the muscle fiber resistant to this genetic problem? And that'll be our therapeutic avenue. Exactly.
That's a treatment.
That could lead to treatment, exactly, if you can upregulate that. And so my hand waving.
All right, Dr. Bonneman, thank you so much. That was wonderful.